Good day, everyone. And I'm Nicoletta La Rocca from the Department of Biology at the University of Padova, and I focus always my research on studies the photosynthetic responses to different environmental conditions. And today we'll speak about the photosynthesis under simulated red dwarf spectra and analogous light niches on Earth. And before starting, I will say that, as you maybe know, many missions in the last decade discover potentially habitable exoplanets, and now two planned missions, in particular HWO, Habitable World Observatory, and Ariel, will try to characterize them in terms of biosignature. Biosignature are a sign of the life presence in a planet that can be recorded and recovered from remote. And they can be both atmospheric biosignature, if they are linked to the release of some gas from living organism, and in particular on Earth we can see the presence of oxygen and ozone in our atmosphere, or can be surface biosignature that can help to distinguish soil to water from water and from living organisms present in the surface. So both these signatures on Earth are linked to the oxygenic photosynthesis. And the oxygenic photosynthesis is due to the activity of photosynthetic organisms and bacteria, algae, and plants that are able to convert the CO2 and water to carbohydrates here, and so in biomass that is present in all the photosynthetic organism, and release oxygen in our atmosphere in huge amount. The photosynthetic process is allowed by the light energy absorption by photosynthetic tissue of plants, algae, and cyanobacteria, and pigments absorb the light and can utilize the energy to build up the carbohydrates and release the oxygen. Nowadays, the photosynthetic organisms represent more than 90% of the living biomass on Earth, and so oxygenic photosynthesis produces a robust atmospheric biosignature, and this is a key target also in the search for life outside the solar system. The other type of biosignature, so the surface biosignature, is also typical due to the photosynthetic organism, because we can observe when vegetation is present such kind of signature with very low reflectance in the visible portion of the light, as photosynthetic pigments mainly absorb visible light, while we have a very strong reflectance in the near infrared due to the reflection by the plant tissue. In between, we had this sharp increase in reflectance that is called red-edge, and this is a fingerprinting of surface biosignature on Earth. And what about the possibility of photosynthetic habitability in exoplanets? Most of the planets potentially habitable have been found around red dwarf, also called M-type of stars, and only few orbiting K stars. In both cases, this type of stars, respect to our Sun, that is a G-type of star, are characterized by a lower luminosity, a lower temperature, and most important, a very different light spectrum. The light spectrum of the Sun, here in purple, is characterized by by very high light contribution in the visible portion of the spectrum, while the other spectra, the spectra generated by the other type of stars, are depleted, more and more depleted in the visible light and reach the infrared. As you can see, looking at the K stars and M type of stars. The M type of stars represent the worst case scenario, as they have very dim light, they generate very dim light in the visible part of the spectrum, while are enriched in infrared and near-infrared photons. And this is challenging because the photosynthetic organisms utilize photosynthetic active radiation, so the visible light between 400 to 700 nanometer to photosynthesize. So starting from this knowledge, we can consider different research questions. Are on Earth photosynthetic organisms capable to exploit M-dwarf light spectra? And how much do they grow and photosynthesize under the simulated M-dwarf light? And could we expect strong biosignature from this type of photosynthesis? To respond to this question, we can have different approaches. So we can study the photosynthetic biodiversity on Earth. And on Earth, we have different light niches and different type of photosynthetic organisms that are able to photosynthesize even in very dim light, like the Dichyophyllus plants that are saturated by very low light intensity in the photosynthetic process. But we have also Heliophilus plants that like much more the light of the sun and that are saturated in the photosynthetic process by higher light intensity. We can use different types of photosynthetic organisms to perform a laboratory simulation to test their productivity under different starlight spectra. And we finally can utilize the data coming from photosynthetic biodiversity studies and laboratory simulation to perform numerical simulation and to better understand the habitability of exoplanets. Speaking about photosynthesis, we have to say that photosynthesis evolved just once, quite early on Earth. And this event completely changed the atmosphere of our planet that became more habitable and completely changed also the life form in our planet. The photosynthetic process was evolved by cyanobacteria early in the Archean, and they were also responsible of the great oxygenation events that happened about 2.2 billion years ago. And after that, the oxygen increased in the atmosphere, in the Earth's atmosphere, and pushed the evolution of most complex form like eukaryotic organism, algae, and plants. And at the end, with all these type of photosynthetic organisms, we reached the concentration of oxygen that we can measure now in our atmosphere, so the 21%. During the evolution of the different type of photosynthetic organisms, so we had different forms, different morphology of the organism, and during the land colonization, we had also the possibility to see the evolution of plants, higher plants, and during that time, different photosynthetic organisms had to cope with terrestrial stressors, different terrestrial stressors, availability of water, humidity, light, nutrients, and so on. So we can observe a very high biodiversity of photosynthetic organisms, but this biodiversity is not only in terms of morphology. The photosynthetic organisms all utilize photosystems that harbor photosynthetic pigments, and the photosystems that are characterized by a reaction center, where that is maintained along all the evolution of photosynthetic organisms, and this is still present in all photosynthetic organisms, harboring chlorophyll A and beta-carotene, is surrounded by antenna system. The reaction center is surrounded by an antenna system. The antenna system that harbor different type of pigments can collect different photons of different wavelengths and transfer the energy to the reaction center to start the photosynthetic process. What was diversified in the different photosynthetic organisms is the antenna system that harbor different type of pigments that could match the light niches where the different type of organism can live. So if we look at the pigments that are present in the different antenna systems, we can find many type of pigments that absorb many different wavelengths. And this is really important because, as I told you, the photosynthetic organism doesn't live generally under a continuum of light spectrum like this, but can experience different light intensities and different spectra. Even in the Earth surface, we could have a very different light spectrum. By the sun, at noon, we have a complete spectrum in the visible with a very high amount of photons, 2,000 of photons more or less in the visible, while in the evening we have less than 100 photons, and the light spectrum is deeply different. And plant has to thrive under the different light spectra, and even in spectra that resemble the M-dwarf one. Like plants that are living in the understory environments. Here, despite the light that is perceived by the plants is depleted in the visible wavebands, while is increasing is reached the infrared and near infrared, like M-dwarf stars. Waters also are characterized by different light spectrum depending on that and on turbidity. And so we can experience also by microalgae and algae spectra that are enriched in infrared. Putting all together the pigments that have evolved by the different photosynthetic organisms, we can see that the entire visible part of the sun spectrum is covered to utilize all light niches on Earth. And the light above 700 nanometers was considered not enough energetic to sustain oxygenic photosynthesis. However, we had some surprise in the last decades, as we can find terrestrial niches analogous of exoplanet-like light, where photosynthetic organisms can live. We can find cyanobacteria in caves or in microbial biofilms or under the sands or in the rocks, where the organism, these photosynthetic organisms perceive a light spectrum really reach the infrared light and very dim and with low emission in the visible, low presence of photons in the visible. And under this environment, cyanobacteria can live and can photosynthesize and release oxygen thanks to special adaptation. In particular, some species of cyanobacteria resulted to be able to photosynthesize even in just infrared light. They can utilize visible light, but if the visible light is not available, they can reorganize their photosynthetic apparatus and synthesize new pigments that absorb at longer wavelengths, so from 700 to 800 nanometers more or less, like the chlorophyll D and F, as you can see here. When they incorporate this chlorophyll, this special chlorophyll in their photosystems, in the in vivo absorption spectrum of the cells acclimated to infrared light, we can observe a change the age of the different peaks due to the reorganization of the photosynthetic apparatus, and we can observe this shoulder that appear above the 700 nanometer that is due to the incorporation of chlorophyll D and F. So we have a very strong biodiversity and high biodiversity also in this term. Recently, a number of microalgae, not too much indeed, have been discovered capable of utilizing infrared light. This type of algae live generally in environments where the light is enriched in infrared light, in coastal waters, turbid waters. We can find this Phaeodactylum tricornutum that is able to utilize infrared light. And also we have some example of algae that live in symbiosis with corals that perceive a light spectrum really similar to the M-dwarf one. And Chromeravelia is the one of the most studied microalgae that can live inside the coral tissue. Also, in the case of microalgae, when they have just infrared light available, we can observe the appearance of a shoulder of absorption above 700 nanometer. But in this case, it was discovered that the algae didn't synthesize new pigment. They just organized the canonical pigments in different environment, different protein environment of the antenna complexes that change their absorption features. And so the same type of chlorophyll A can absorb at longer wavelengths, and is called redshifted chlorophyll A. So the proteic environment can also change the characteristics of absorption of a pigment. And then we have Staphylos plants, so plant that is living on the story and is adapted to very low light and with very low level of photons in the visible and least infrared. In this case, we can observe also a redshifted chlorophyll A phenomenon with the appearance of not a shoulder light, but just a portion of the spectrum that is able to absorb about 700 nanometers. And in case of Staphylos plant, we have also many structural features that help plants to photosynthesize under dim light, dim visible light, and infrared light. And they can have conic epidermal cells that can harvest, that can collect light coming from a different direction. And they can focus, thanks to these conic epidermal cells, the light to the photosynthetic tissue. And this could help in utilizing more light by this tissue and these cells. And they also show very big chloroplasts, giant chloroplasts, so the photosynthetic organelles inside the leaf tissue. And in these giant chloroplasts, we can also observe a particular multi-layer membrane structure that has a very ordered organization, and it has been demonstrated that this multilayer structure can have a photonic effect and enhance the use of infrared, dim, visible, and infrared light by this plant. So at the end, infrared light is less energetic, but can drive oxygenic photosynthesis in organisms with unique physiological and structural adaptation. So now in the book, we speak about extended power. And so we consider the photosynthetic active radiation from 400 to 750, 780 nanometers. Starting from this knowledge, we perform laboratory simulation and laboratory screening of photosynthetic organisms exposed under M-dwarf light spectra. We consider organisms with different complexity, from cyanobacteria to higher plants. And to perform this laboratory screening, we utilized a facility that is now present in our lab, in my lab, in the Department of Biology of Padova. But this setup was built up in the last 10 years, thanks to a collaboration between our lab and the Institute of Photonic and Nanotechnology in Padova and the National Institute of Astrophysics, and in particular Riccardo Claudi, that started with the idea to build up a starlight simulator. Now the setup is much more complex than at the beginning, and we have light simulators that are combined with the atmosphere simulator chamber, where the organs can be grown under the light source that we decide to apply, and under a specific atmosphere. And the atmosphere simulator chamber that hosts the organism that we want to test is exposed to the light of the stellar light or different light simulator. And in this chamber, the organism growing increase the oxygen in the atmosphere, and the changes in oxygen level and the CO2 use can be detected from remote by special sensor that allow to measure online the photosynthetic activity of the organism. And then the setup can be also equipped with a reflectance detection system that allow to collect data about the reflectance spectra of the organism growing under the different conditions. The starlight spectrum is an illuminator of about 280 LEDs in 25 channels. They can be tuned to reproduce the light spectrum of different stars. Here you can see the spectra of the M7 type of stars, M-dwarf stars. Here in blue, the generated spectrum. In red, the smoothed spectrum generated from the starlight spectrum, here in gray. And so the reproduction of the light spectra are really reliable, and yeah, they can be reproduced. They can reproduce the starlight spectra with very high reliability, up to 850 nanometers. The atmosphere simulator chamber that allow to grow the organism in a certain atmosphere are also equipped by sensor to detect the changes in CO2 and oxygen level during the experiments, and so allow to evaluate the impact of photosynthetic microorganisms and organisms on the atmosphere composition when exposed to the different starlight and non-terrestrial atmospheres. Now we have, beside the starlight simulator and the atmosphere simulator chamber, we have other two setups to simulate just solar light and associated with an atmosphere simulator chamber, or monochromatic infrared light, always associated with the atmosphere simulator chamber. In this way, the experiment speed up much than in the past, and so this allowed us to do a lot of work. And I don't want to bore you, but if you are interested in the biological data, you can look at our publication. In all cases, when we simulated the light spectra and we compared the responses to solar or to infrared or to M-dwarf light spectra in the different organisms, by the different organisms, we utilize the same photon flux density, and we maintain the same amount of photons, while the photon distribution was different in the different wavebands, and so the total energy changed depending on the light spectrum. As you can see from this table, the percentage of the visible light was strongly different in solar simulated light, M-dwarf light, or infrared light, while the infrared portion of the photons accounted for higher amount going from solar to infrared light. And we tested different atmosphere, terrestrial atmosphere and anoxic atmosphere, made by 90% of nitrogen and 5% of CO2. And with this spectra and atmosphere, we tested at first cyanobacteria. And at the beginning, we considered two types of cyanobacteria. One species capable to utilize just visible light, and a second species capable to utilize visible light or infrared light, or both when both were present. And we analyzed their photosynthetic performances, and we saw that in both the organisms, the photosynthetic activity under M-dwarf starlight was quite high, and so quite efficient, and not comparable with the activity under solar light, but near the activity under solar light, the photosynthetic activity under solar light. And so it is possible to perform photosynthesis and to release oxygen under M-dwarf starlight. And during the acclimation of the organism to the different light, we also observe changes in the composition of the pigments. And in case of a possibility by the organism to utilize infrared light, we observe the appearance of a shoulder above 700 nanometers. The shoulder was much higher, much bigger in the infrared acclimated organism, while was less pronounced in M-star light, under M-star light, but anyway present. And the changes in light absorption by the cells obviously could affect the reflectance spectra of organisms that could live in exoplanet. If we have, as an example, a selection of a type of organism that can utilize infrared light, we will observe a change in the reflectance, so in the red edge, in particular due to the absorption by chlorophyll DNF and infrared light. So it appeared that the species capable to utilize infrared light besides visible light had some more chance to photosynthesize and release oxygen under M-dwarf spectra. But when we analyzed another type of organism, also able to utilize infrared light, but generally living in very low visible light, we observed that this oxygen release was less than expected. And by analyzing the in vivo absorption spectrum of the organism exposed to infrared, M-dwarf, or solar light, we observed that this type of organism that use very low visible light is able to use very low visible light, prefer to acclimate the photosynthetic pigments that absorb the very low visible light available in M-dwarf spectrum, instead to synthesize new pigments. the new pigments were synthesized just under far-red light, but in the mixed spectrum with very low visible light and far-red enriched of an M-dwarf stars, chlorophyll D that aren't synthesized by these organisms. And so we can say that at the end, depending on the organism, we can have different responses under M-dwarf starlight. This organism also showed to change its color depending on the light that was perceived. And so we cannot generalize and say that far-red users are more able to live or to photosynthesize under M-dwarf simulated spectrum. So the final results will depend on the reorganization or synthesis of new pigments, and this could obviously affect the reflectance spectrum. Then we move to microalgae. We utilize also in this case visible users and visible plus far-red light user. And we consider Chlorella vulgaris, that is a freshwater organism, and Nanochloropsis gaditana and Chromarabellia. Nanochloropsis is living in coastal waters, while Chromarabellia is a symbiont, as I told you earlier. And we analyze the capability of this algae to grow under the different light spectra, and we observe that algae that has the possibility to use red light beside visible light had a much faster growth, a much efficient growth under M-dwarf spectrum with respect to the solar one. Respect to the far-red one and near to the solar one. So it seems that in the case of algae, the far-red users has some advantage respect to the non-far-red user that grow less. This paper is under review, so please don't don't utilize the data. And then analyzing the characteristics of the in vivo absorption spectra of the algae, we saw that Chromarabellia was shifting its chlorophyll A to absorb light above 700 nanometer. This red shift was more pronounced under far-red light than under M-dwarf light, and so there is an activation dose-dependent of this red shifted phenomenon of the chlorophyll A. While the other species able to utilize far-red light doesn't show a change in the spectrum, absorption spectrum above 700 nanometer, but it seems to have a constitutive tail of absorption above 700 nanometers. And together with this trait, Nanochloropsis gaditana showed to have an increase, very high increase in the synthesis of photosynthetic pigments, and in particular of chlorophylls, and also showed in cells acclimated to far-red light, showed very big chloroplast. So again, giant chloroplast with very well-organized membranes, huge amount of membranes, and we think and we speculated that this could have a photonic effect similar to that we observe in plants living under storey environment. Then we also consider plants, vascular and non-vascular plants. We analyze the responses of a moss, modern moss, Physcometrium patens, to the different light spectra, and we observe the reduction of growth under M-dwarf light with respect to the solar light, the moss wasn't able to grow under infrared light, obviously. And it was also interesting to observe that under M-dwarf light respect to solar light, delayed reproductive structure development was observed. Even if the photosynthetic activity was similar in the tissue present under the moss under the two different light spectrum. And finally, we analyzed the responses of Arabidopsis thaliana, model plants, an Heliophilus plant that like very much light, so not understory plant. And in this case, we observe that photosynthetic tissue maintain quite good photosynthetic efficiency, but the growth of the plant is not normal, is altered with the petiole of the leaves that are quite long, and the plants that grow in a different way with respect to the plants growing under solar light. And this is a typical response to shade avoidance by Heliophilus plant. And so this type of plant under M-dwarf star, we could have difficulties in the development. So to summarize this part, we can say that cyanobacteria has a strong growth and normal development under M-dwarf spectrum, and they can work very well, both in case they are able to utilize or not utilize the infrared light, and they resulted similarly efficient in using infrared light, M-dwarf light. The microalgae also showed a strong growth and normal development, and non-infrared light users seems disadvantaged by respect to the infrared users in using M-dwarf light. In the mosses, we had lower growth and the altered development, and also in the Heliophilus plant. We never studied until now Schiaphilus plants, so plants that have specific adaptation to low visible light and infrared spectra. So this could be a point for the future. And then I have some tips about the photosynthetic habitability under M-dwarfs. It is important to accurately simulate the M-star spectrum, otherwise we can underestimate the photosynthetic habitability of exoplanets. So the low amount of photons in the visible are determinant to allow the photosynthetic organisms to evolve oxygen, to all the photosynthetic organisms to evolve oxygen. Under the simulated M-star spectra, the oxygen evolution and growth potential of all the phototropes is quite efficient and can impact even anoxic atmosphere. We did experiment with the anoxic atmosphere with just cyanobacteria, but anyway, we will test also microalgae. The photosynthetic organisms use different acclimation strategies to infrared spectra that could differently influence the in vivo absorption spectra and thus the reflectance spectra and the red age. And the photosynthetic biodiversity of visible plus infrared light users is surely underestimated. So studying the biodiversity of photosynthesis is really important. And combining simulation with photosynthetic biodiversity study, we can better evaluate the exoplanet habitability. And finally, we decided in the last 2 years, we had a grant, and we decided to implement our setup with a simulating stellar flare equipment. And so we also decided to simulate stellar flare at the planet surface. So stellar flare attenuated by anoxic atmosphere, we build up this UV flare simulator, and that is made by 12 different wavelengths, LED of different wavelengths, organized in 10 channel, adjustable through custom-made software. And here in the lower part, you have an example of the 3 gigayear sun flare filtered by a potential Archean atmosphere. In green, the mathematical model, and in red, the reproduced spectrum of the flare. And so we want now to combine the stellar spectra plus the UV flare, the anoxic atmosphere, and to understand the growth capability and physiological responses of desertic and hydrothermal cyanobacteria, capable of use infrared light beside visible light, and also capable to utilize to synthesize UV protecting molecules. So this is our final goal, and we will see the capability of this extremophile organism to go with all these conditions. Finally, I would like to acknowledge and thanks the research that are working or that work in my research group and lab, and in particular Mariano Battistuzzi, that some years ago did the PhD in my lab, and then 2 years of postdoc, and Elisabetta Listro, that is now postdoc. Beatrice Boccia is a PhD student, Anna Segalla is a technician, and Thomas Morosinotto is a colleague. And also Luca Poleto and Lorenzo Coccola from the Institute of Photonic and Nanotechnology in Padova, Riccardo Claudi from the Astronomical Observatory in Padova, Daniela Billi in the University of Roma Tor Vergata, and Laura Silva and its team, Michele Maris and its team at the Institute of Astrophysics in Trieste, and the Italian Space Agencies for the funding. And thank you for your attention. And finally, if you are interested in this topic, you can follow the extraterrestrial photosynthesis workshop that will be on middle of May, and that can be followed by remote. So, thank you very much, and I'm open for questions. Think about an oxygenic microbial photosynthesis as it applied to red dwarf. Yes, obviously, an oxygenic photosynthetic organism can be utilized to test their capability to grow under this type of spectra, but I do expect that they will grow because we saw that cyanobacteria and also microalgae are able to grow under this type of spectrum. So an oxygenic photosynthetic microorganisms that are able to absorb longer wavelengths can have a very good chance to grow under M-dwarf spectrum. Anyway, we cannot utilize the sensor to detect their photosynthetic activity. We are just, in this case, to understand if they are able to grow by measuring other type of parameters. And also some anoxygenic photosynthetic bacteria cannot grow under oxygenated atmosphere, so it's not so easy to handle them and to perform the experiment in normal environments and to utilize this equipment. Just the bacteria, the anoxygenic photosynthetic bacteria that can cope with oxygen could be easily utilized, but this is a good point anyway.
Instead of simulating the full spectrum of the M-dwarf light, if you just give the organisms cut out the visible light with some filters and only give far red light and see what.
We didn't try until now, but this is a good point anyway. We can better understand if the organisms are just utilizing the visible light or the combination between low visible light and infrared light lead to the organism to grow better. This could be because we have the Emerson effect that can push the activity of one of the two type of photosystems and help in the photosynthesis, the organism.
Yeah, well, it'd be interesting because they're probably using the far red light to boost the visible photons.
Yeah,
or
yeah.
Interesting.
We said something like this in the last paper that is under review now, because in algae, if I can share again my video. In the algae, we have a very strange unexpected response. So, this one. Okay. In Nicoletta gaditana, that is an algae that is able to utilize infrared light, but less efficiently than Chromeravella, that you can see here can grow much faster under infrared light respect to Nicoletta gaditana. We observe a very fast growth under M-dwarf spectrum. So as this type of algae has a constitutive tail of absorption in the infrared, doesn't need to acclimate to infrared light to use it. Respect to Chromeravella, we observe this burst of growth, and this could be dependent on the effect of infrared light on the entire photosystem structures and functionality. And so this could be, and this is a point, but you have to think that we take many days for each experiment, each test to be done in triplicates, quadruplicates, biological samples, and so on. So it's not so easy. But thanks for the question.
Thank you, Nicoletta.